If you remember from last time, we were covering kind of the basics of photosynthesis in terms of the reagents and the products, and we broke down photosynthesis into two sections, the light reactions and the Calvin cycle. So we're going to cover both of those today. So just want you to focus in on the left. You can see we're talking about using light and water. We're producing oxygen, ATP, and NADPH. By the way, if anyone has any questions, you can put them into the chat feature for Zoom, and I'll try to check that from time to time to see if there's any questions. Or you can always speak up as well, and just look for an appropriate time to interrupt me. Or if I'm going too fast, let me know. Sometimes I know I talk a little fast. All right, so yeah, I showed you this slide. NADP plus and ADP are recycled and reused. And the whole idea is we're starting with light and making sugar. So I finished off with this slide here last day. And I know I had zoomed through it. And what I'm going to do is kind of just pick it apart here today, kind of step by step. And so this is the system in plants. When you get to algae and bacteria, sometimes it's a little different. You can see there's actually two photosystems in plants. Some bacteria have one or two. The confusing thing, unfortunately, is that the first one is photosystem 2 and the second one is photosystem 1. Unfortunately, that was just the order they were discovered historically. So the second photosystem was discovered first, and then when they discovered a new one, so anyway, sometimes they're called photosystem 680 or photosystem 700 because those are the wavelengths that they absorb maximally. So take a look at this. Here's our inputs. We have our photons over here. We have our water over here. We're making ATP and we're making NADPH. So what I want to do is kind of go through this step by step and take a look at this. And I'm going to write some notes. And I know I have notes in the PDF files that I provide you, and it's going to be kind of similar to what I'm writing. But I find it best to kind of just break this down one step at a time and write these notes. Otherwise, we zip through it too quick and it's a little hard for people to follow. So let's take a look at this here. So you can see the first note there. In the first note, we basically have, so we have light. So let me write down what's going on here. Okay, so we have light hits chlorophyll. And I actually can't remember if this is chlorophyll A or chlorophyll B. It doesn't really matter. So light hits chlorophyll. So that's kind of the first step there. And what happens is that energy is transferred to electrons in chlorophyll. And so electrons from chlorophyll, they're basically transferred to that reaction center, which is called P680. I'm going to kind of skip over that and basically say what happens to those electrons. And those electrons are donated. Electrons from chlorophyll are donated to an electron transport chain. So I'm just going to put ETC for electron transport chain. So that's the first step. Energy comes in, it rips electrons off chlorophyll. Those electrons are put into an electron transport chain. And we'll get to that in a second. So what is number 2? Number 2 is water. Oh, sorry, I forgot this is an extra slide. Hopefully everyone got that. You can always go back and replay the video. So number 2 is what do we do with those lost electrons? We want to replace them. And this is where water comes in. So number 2, or actually maybe I have these steps mixed up. Sorry, this is number 2. Number 3 is the water part. Apologize for that. I'm just trying to, I know I changed these at one point because I realized that the steps I had were slightly different from the textbook. And I'm always trying to line things up with the textbook because I know that's a major resource for everyone in the class. So 1, light hits chlorophyll, electrons get excited. Number 2, the electrons are ripped off chlorophyll and donated to an electron transport chain. All right, so number 3, I'll change this to a 3. That doesn't look like a 3. Let's try that again. There is a 3. Okay, so number 3, water is split. All right, so this was the reaction that I wrote on the board before. So H2O, it's broken down. and we get 2 hydrogens plus 2 electrons plus an oxygen. And just to balance the equation, it's 1/2 a mole of oxygen. I know some people like balancing equations, and so that's something I like to do just to keep track of everything. All right, so, sorry, don't know what happened there. All right, so some notes about these things, okay? So the oxygen, this is a byproduct. Byproduct. Okay, sorry about that. I'm just cramming things in a bit too much. The electrons, these electrons, they replenish the lost electrons in chlorophyll. So lost electrons in chlorophyll. Okay, and then the last are the hydrogens, and the hydrogens actually are pumped across the membrane, and they become part of the proton motive force. So we can put that pumped across thylakoid membrane, and creates proton motive force. All right, so hopefully you get all these acronyms here. So that's number 3. So at this point we have water has come in, it's stripped some electrons off of chlorophyll. The electrons are going to electron transport chain. We're going to make a proton motive force here in a minute. What is the role of water? Water is replenishing those lost electrons from chlorophyll. So that's 1, 2, and 3. All right, so number 4 is really just the electron transport chain. So number 4, oh, what's going on here? Okay, number 4 is the electron transport chain. So number 4 is we have energized electrons are used to pump protons. And so they're being pumped into the thylakoid space. So maybe I'll put that into thylakoid space. And like we said already, this creates proton motive force. All right, so, and what do we do with the proton motive force? Pretty much every time we have a proton motive force, we're rotating something. It could be a bacterial flagella, or in many cases, really, we're just making ATP. All right, so proton motive force is used to make ATP. So same way ATP is made in the mitochondria, we could talk about ATP synthase. So maybe I'll write that down. ATP synthase is the enzyme that does the whole thing. So that is useful. We have some ATP. Somehow in this whole process, we've taken energy from the sun and we've put it into chemical bonds in ATP. But this is really just a small part of the whole thing because, like I said, ATP is not useful for storage, and so we don't want to do a little bit more. And this is where the second photosystem 1, comes in, is that plants are a little extra efficient at doing this kind of thing. So take a look, number 6, we have light hitting chlorophyll again. Okay, so we have light hits chlorophyll. Light hits chlorophyll, and I think this one is chlorophyll B, but I can never remember which is which. So I'm just going to put chlorophyll. And in this case here, we have electrons are going to get promoted again. So we'll say that, and energizes proton, or energizes electrons. Energizes electrons. And ultimately, this happens in the second reactive center. So I'm going to put at photosystem 700. All right, we're almost there. I know this is a lot of steps, and I will not ask you to regurgitate all of these steps. Like I said, when I get to the end of this, I want to give you a good idea of what's expected for the midterm. So we'll get there. All right, 7 and 8, we have another electron transport chain. So we can say that 7 electrons are used in another electron transport chain, ETC. And then number 8 is what happens to those electrons ultimately, okay? So we can say that electrons become bonds in the reduction of NADP, in the reduction of NADP plus. Okay, so here's the chemical reaction, right? We have NADP plus. We have 2 electrons because those are, of course, going to become a bond. We get the balanced equations, we actually get 2 protons. And then the reactants, we get NADPH. And one of the protons, as part of this, actually gets pumped across the membrane into the thylakoid space. Kind of the key thing here is we've made ATP and we've made NADPH. And that's kind of all that matters about the light reactions, is we're making these 2 energetic products because cells like energy. And so now we've captured light energy into new chemical bonds, and this is very, very useful. And this is what the light reactions are all about. So 8 steps, like I said, I'm not going to ask you to regurgitate all 8 steps. And in a minute, I'll try to explain to you what I think is the most important about knowing about photosynthesis. Here's an image from the textbook. It's trying to give you an idea of kind of how this works in terms of thinking about potential energy, right? So remember before we had the idea of the diver jumping into the water. In this case here, you can see you've got this guy is representing a photon hitting that electron with a hammer and is driving up to a higher energy level. The higher energy level is used, some sort of a windmill to make ATP. That happens again, and the electrons get promoted and eventually end up in NADPH. So bottom line is we have light going in, and we have ATP and NADPH going out. The water and oxygen are great too, because we like oxygen, but for the plants at this point, the oxygen is not really that important at all. It's just a byproduct. So here's a kind of a little snapshot of the thylakoid, right? And you can see up there on the top, you have the chlorophyll. The chlorophyll is right in the membrane of the thylakoid. That's the green part of the plant. And you can see that you've got these protons here. As the light hits, as the protons are getting pumped, we're making a proton motive force. Here's our ATP synthase down here. And ultimately, we're making ATP. And the electrons are ultimately also ending up on NADPH. And notice where they are going. They're ultimately going to the Calvin cycle. All right, so like I said, that's kind of the important thing to know for the light reactions. What are those final products? ATP and NADPH. Okay, so kind of a teeny tiny footnote, which I will never ask you about on an exam. I just want to distinguish between, remember these previous ones here, these previous slides. Notice it's called linear electron flow. And if you go in the textbook, it kind of talks back and forth about the cyclic and the linear. So I just wanted to clarify what's going on. It turns out for the Calvin cycle, we actually need quite a bit of ATP. And so what actually happens in cells, rather than the electrons going from the beginning all the way to the end, is sometimes they're cycled back to that first electron transport chain to make extra ATP. Because it turns out extra ATP is good and it's needed for the Calvin cycle. I won't ask you about this on the midterm. I just wanted to kind of clarify that if you're reading about it in the textbook. So there's our ATP synthase. It's the exact same one that we have in mitochondria. The only thing that's different here are the labels. If you remember the mitochondria up here, we had the intermembrane space and the matrix. Now we have the thylakoid space and the stroma, but it's the exact same protein, doing the exact same thing, making ATP. Here's a little comparison. You can see the gray space is where the The dark gray is where the proton motive force is, and the light gray is where the protons are moving towards and where the ATP is made ultimately. So in the mitochondria, the ATP is made in the matrix, and the chloroplast, it's made in the stroma. And that is where the Calvin cycle is going to happen. So there are the light reactions. I keep coming back to this slide. I keep telling you this is the important thing to know. This is really important to know this slide. So do not forget it. So let's take a look at this Calvin cycle. This Calvin cycle, we're going to use these products here, the ATP and the NADPH. We're also going to throw in some carbon dioxide, and we're going to make some sugar. So let's talk about how that's going to work. So let's talk about the Calvin cycle. So just a little joke there with Darth Vader. Sometimes textbooks used to call them the dark reactions, which is not really true. You don't need dark for the Calvin cycle. More accurately, they're light independent. Most people are calling this thing the Calvin cycle because it was studied in great deal by Calvin. Sometimes people talk about Benson and Bassam, which were, I don't know who they were. They probably added to understanding, or maybe they worked with them. I'm not entirely sure. Most people call it the Calvin cycle. Some textbooks are moving towards this term here, carbon fixation, because it's actually describing what is going on. We're fixing carbon. We're taking CO2, carbon dioxide, from a gaseous molecule and putting it into organic compounds. And so it's a little bit more accurate what we're doing. So at this point, I usually in class ask people, you know, if your last name was Kelvin and you know, you had a child, what would be the worst possible name you could give your child? And because poor Calvin here has it, or at least in my mind, it's kind of funny. So let's see what his name is. There it is, Melvin Calvin. I just think it's a really funny science name. Poor guy, but anyway, he overcame his difficulties and ultimately became a famous scientist. There he is. So this is way back in the 1940s, and the lucky guy was working with radioactive carbon dioxide. I say lucky sort of in being sarcastic. Working with radioactive gases, yeah, that's not really a fun experiment to do, and can be quite dangerous. At least though, radioactive carbon is not high energy radioactive. So he was feeding them radioactive carbon dioxide and then isolating the metabolites using a procedure called paper chromatography. And it's similar to thin layer chromatography if you do that in organic chemistry, if you take that next semester. So let's take a look at the Calvin cycle, okay? There are a bunch of interesting and long-named chemicals in the Calvin cycle. And it really starts off with this one here. And you can see the stoichiometry that I'm not sure why, but the enzyme uses 3 and 3 of each. So 3 carbon dioxide over here and 3 of this thing here, RUBP. The full name is ribulose 1,5-bisphosphate. Bisphosphate means 2-phosphate, like 5. And it's a ribulose, it's actually a 5-carbon sugar. And what it does, it's going to combine these 2 things, and it's going to, there's kind of a short-lived 6-carbon intermediate, and ultimately makes 3-phosphoglycerate. So some of you eagle-eyed people are probably thinking, wait a second, I recognize that. There was a phosphoglycerate in glycolysis, and you're right. This is actually a very similar compound. But you don't need to know the details for this. I would know this one here though, RUBP. If you want to know the full name, that's great. But it's sufficient if you just know RUBP. The other thing to know is the enzyme that does this reaction. Some people have said that this may be the most important enzyme on the planet. So ribulose 1,5-bisphosphate carboxylase. So that means it takes the RUBP and adds a carbon dioxide to it. So you don't need to know the full name, but you should know Rubisco. And I can tell you last year on the midterm, one of the short answer questions was, what is Rubisco? And A lot of people didn't know. I even told them, know the name of this enzyme. It's very, very important. I'll even put a big star here for you. I'll put a second star. Here we go. There we go. A couple of stars. I can make it a 3-star if you want. That will give you the hint. It will be on the midterm somehow. So make sure you know what it is, okay? And so if you want to define it, you could say this is the enzyme that fixes carbon. This is the enzyme that takes carbon dioxide out of a gaseous form and attaches it to RUBP. There's lots of ways we could describe this enzyme. I'll show you, I've got a picture over here. There's the enzyme on the left, and you can see on the right, it's been fluorescently labeled. It turns out it's actually a membrane protein, and it's shown there in the chloroplast. And like I said, the most important enzyme on the planet, because this is how we get carbon dioxide from a gaseous form into organic compounds. And so a massive part of the biosphere is reliant on this enzyme ultimately. All right, so back to the textbook image, and you can see this is basically the exact image I showed you, just kind of maybe a different version. They're not showing the chemical structures, they're just showing the carbon as these little gray balls. And so we start off with RUBP, which is 5 carbons and 2 phosphates, and we end up with a 6-carbon intermediate, which becomes these 3 carbon, 3 phosphoglycerates. And so there's a few more things that are happening now, right? You can see this thing here, it starts off and it has phosphates on it, so it's energized. So that compound is already primed and energized. And so when it's broken down into 3 phosphoglycerate, it's ready to start even absorbing more energy. And so you can see we have some ATP going in. So electrons from the ATP are changing this thing to another compound that has higher energy bonds. And this happens again, except for now with the NADPH. You can see it gets recycled to NADP+. And ultimately, we get this thing here, glyceride 3-phosphate, so G3P. So if I ever ask you, what is the product of photosynthesis, this is it. So this is the product of photosynthesis, I know everyone wants to say glucose is the product of photosynthesis, but it's not. It is in some cases. It depends on the plant. Some plants take glyceraldehyde 3-phosphate and they make other sugars. So they might make fructose, they might make glucose, or they might make starch. There's other things that can be made with that. So G3P is a good building point. I'm not sure what percentage, probably 90% of the time it's used to make glucose, but it is the technical product of photosynthesis. So just so you know. So this is a cycle. It's not quite done yet, and so some of the glyceraldehyde 3-phosphate goes on and is going to will make some organic compounds. Some of it gets recycled, and it ultimately it takes a bit more ATP over here, and we're going to regenerate the RUB. So that's the Calvin cycle. A lot more detail in the textbook. Don't want to get bogged down in the details. Kind of want to just think about what is going on here, right? We're taking carbon dioxide. We're taking ATP, we're taking NADPH, and ultimately we're making this thing here, this glyceraldehyde 3-phosphate. So that is really what I want you to know. Obviously, there's some recycling going on, like the ADP and the NADPH. These are going to the light reactions. They're going to get recycled, and that's part of the bigger picture of photosynthesis. So there is the very basic version of this. And like I said, a few names in there to know: Rubisco, RUBP, G3P, or glyceraldehyde 3-phosphate. You can know the acronym, I'm fine with that. And don't forget this enzyme, Rubisco, most important enzyme on the planet. Okay, so depending on where we're at in terms of the getting close to the midterm and things like that, sometimes I spend a bit of time reviewing photosynthesis now. I'm not going to do this here. I'm actually going to save this for next week when we have our review sessions, so that you've had a bit of time to digest photosynthesis and you know, kind of sleep on it. And so I'm going to come back to this next week. and look at these questions here. You can see I have two questions. One, list the reactions and products of the two parts of photosynthesis. And the second is kind of comparing what is going on with respiration and photosynthesis, but at a molecular level. So comparing the electron transport chains and ATP synthesis and thinking about what is going on with these two processes. And like I said, I'm going to come back to this next week. If you want to work on that, that's fine ahead of time. Might help you with your studying, but I'm going to focus on this a little bit more next week, this review part. This is showing the two parts, by the way, right? Remember the light reactions are on the left, and the carbon fixation or the Calvin cycle is, of course, on the right. And this is a little bit more of a complete picture. I want to talk a little bit about leaves and plant physiology just for a second here to kind of fill in some of the details there. You can see a couple of things are going on here. Where does the water come from? Well, the water is, of course, absorbed by the roots. So you probably know that when you water a plant, you can put water on the leaves. That's not really that useful to the plant. You want to put it in the dirt around the roots because that is where the water is absorbed into the plant. The other thing you can also notice here is it's showing that the gas is exchanged at the leaf, and we're going to talk about that in a minute. So carbon dioxide is going to go into the leaf, and oxygen is going to leave the leaf. And the last part of this picture, of course, is the product. You can see they're talking about sucrose being made, and it will go into the veins of the leaf. And my brain just did a little brain blip here. I can't remember if it's the xylem or the foam. Yeah, I can't remember. My brain it went blank. But it goes down the vascular system, and in some plants, it goes to other parts of the plant body and is often stored in the roots in many types of plants. Let's take a little closer look at the leaf here in a second. There's the diagram in a bigger form. I told you I was going to come back to this leaf cross section, and you can see we have these cells in there, and these cells are there's lots of space in there, so they're kind of spongy, and so they're actually called a spongy mesophyll, this part of the leaf. The top of the leaf, again, depending on the plant, there's many different types of plants, but often top of the leaf has a bit more of a waxy covering to it that kind of, the whole idea is to prevent extra water loss from the leaf. We don't want our leaves to dry out, but it's transparent and allows photons to go in, allows light to go in. At the bottom of the leaf, there are these pores here, and these pores are called stoma. So remember, I told you the inside of the chloroplast is the stroma. So this is not the stroma, this is the stoma. I apologize that we have different terms here for two different things that are very similar, but again, I didn't name them. Stoma is kind of a standard term that means a hole or something like that. If you had many, it would be a stomata, right? You may have heard that term as well. So these little stoma are actually surrounded by these two cells. These two cells are called guard cells. And these guard cells actually have the ability to open and close. And again, it depends on the plant type. There's different stimuli, but if it gets too warm, the problem is the plant is going to dry out, and the plant doesn't want that to happen. So what it can do is actually close a stoma by having those guard cells change their shape. And that's going to prevent water loss, which is an important adaptation. There's many types of adaptations with photosynthesis. If you look in the textbook, it's going to talk about C4 photosynthesis, and there's a few other different types of photosynthesis that are different adaptations in different plants for water loss. And we're not going to get into all that. Too many details that we're not too concerned about in this course this semester. There's some more pictures of these guard cells. You can see these red things are actually the chloroplasts. They're using some sort of a red fluorescence here, and they can open and close. They kind of look like weird eyeballs is what I think they look like, and not a bad analogy because our eyes can open and close too. So there they are. There's another image of them, and all the mesophyll, and you can see all the nuclei are actually nicely stained in this particular cross section of the leaf. All right, so we're almost at the end here. I want to play this video for you, and you're going to see she's going to go through photosynthesis really fast, but you're going to hear her talk about G3P, NADPH, NADP+, RUBP, and so on. And you're going to see all this process happening here. And then afterwards, I have a Kahoot, and then we'll be done early today. So let's play this video for you, and hopefully it can put a little bit of sense into the photosynthesis for you. So we'll give this a go here. Hold on, I'm just going to make sure I actually have my sound sharing. Okay, here we go.
Plants provide us with food to eat and oxygen to breathe. They perform this amazing feat by the process of photosynthesis. Let's take a closer look. Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The two green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the two photosystems. Notice the small mobile electron carriers that shuttle electrons from one large complex to another. Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced two energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With three simple ingredients, carbon dioxide, water, and light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.
Okay, so providing your fuel for studying biology, right? So I have a Kahoot for you here. I'll set this up here in a second. This was just another image I found that shows the stages of photosynthesis kind of integrated with cellular respiration. And if you find it a useful study tool, it's a lot of information packed in there, but I think it's a lot of good information. So take a look at that PowerPoint if you find it useful. If you don't find this one useful, then take a look at the notes that I was putting on the board. All right, so load up your Kahoot app and we'll have a little game and then we'll wrap her up for today.